Hello all. Today I want to talk to you about mitochondrial DNA donation and the concept of three-parent babies. The reason we want to talk about this is because this has been uh, seen in current affairs, and if you look at the bottom of your screen, this Hindu article shows an article published in September 2016. There's another article in 2018, and there's another article in 2019. And all three of these articles in three different sources talk about the same thing: mitochondrial donation therapy. So before we understand what mitochondrial donation is, we need to understand the background of mitochondria. I want you all to go back to your high school biology or middle school biology, where you learned about cell and the parts of the cell, and you may remember learning about mitochondria as the powerhouse of the cell, right? So what does it mean to be a powerhouse? It's because of its function in generating ATP. So the mitochondria is the only place where ATP is produced by a process called oxidative phosphorylation, and this ATP can be used for other reactions occurring inside the cell. So it's almost like a currency, right? You take your currency from one place and you spend it somewhere else. It's the same way ATP is produced in mitochondria, and that ATP is used in other reactions all over the cell. The cell has the most important part of the cell is the nucleus. You can see in this diagram, this blue color region, it's supposed to be the nucleus, and this brown color smaller part is supposed to be mitochondria. There are other parts of the cell too. These are the endoplasmic reticulum. Uh, there are ribosomes and so on. So let's stick to just nucleus and mitochondria for the time being. The mitochondria is also unique because it is the only other organelle other than the nucleus that has DNA. So what's the difference between the DNA in the nucleus, and what's it between the DNA in the nucleus and the one in mitochondria? But for starters, the DNA in the mitochondria encodes only 37 genes. The nuclear DNA encodes more than 20,000 genes. So this nuclear DNA is what you get from your mother and father. Okay, but this mitochondrial DNA is what is already present in the mother cell. So that is the most important information that we have to remember. The mitochondrial DNA is believed to be inherited only from mothers. So how does this happen? If you look at the diagram to your right, you have this father and you have mother. The mother produces the egg, the father produces the sperm. And if you look at this egg cell, you have a nucleus and you have many mitochondria. Most cells have more than one mitochondria. It's not just a single mitochondria as we may have imagined. So in this scenario, the mitochondria of the mother is inside the cell. But if you look at the sperm cell, the mitochondria is just outside the head of the sperm. When fertilization happens, only this nuclear DNA of the father enters the egg of the mother. So that is why the mitochondria of the father does not affect the embryos. So which means we only get our mitochondria from our mothers. So this is what happens during normal fertilization. But what happens if mitochondria is defective, right? So when mitochondria is defective by some means, if it happens to a father, then it's not a big issue because the father's mitochondria is not passed on to their offsprings. But if a woman has a faulty mitochondria, she is going to affect her sons and daughters, right? If this mitochondria is faulty and if she gets married and she has children, all her children will have defective mitochondria because all their children inherit the faulty mitochondria directly from their mothers. So this is why this is a big issue. What happens if the mitochondria is defective? Mitochondria is defective. There is even difficulty in conceiving babies. You can imagine that a cell needs energy, and a growing embryo needs more energy, and even after fertilization, that embryo is not going to carry on to become a very full developed baby. So there may be a lot of miscarriages, and there is another syndrome called Leigh syndrome. It happens in children. So this happened after the birth, and most of those children, die below 10 years of age due to neurodegeneration. And there are many other diseases due to faulty mitochondria, such as Leber's hereditary optic neuropathy. You can imagine that it's an eye disease. It's going to affect vision of children. So there is plenty of serious consequences due to defective mitochondria. And this is precisely the reason why we have to do something about it. So here is what we have come up with: mitochondrial donor therapy. There are there's pretty much two methods to do this to obtain the same uh, result. One method is called the maternal spindle transfer, another is called the pronuclear transfer. Uh, don't try to complicate too much remembering these concepts. There's only slight difference in where the fertilization happens. So let's look at the maternal spindle transfer first. Here the maternal here the maternal cell, which has the abnormal mitochondria, this is taken and her nucleus is removed. All right, her nucleus is removed and this cell is discarded. So we just want her nucleus because we don't want her faulty mitochondria. And we get a donor woman with a healthy mitochondria, and we take away her nucleus and discard the nucleus. So we have the nucleus from the patient and we have the cell from the donor, and we take the nucleus and put it inside the donor. The donor is also a woman and the nucleus is also from a woman. So here we now have an egg with healthy mitochondria from the donor and the nucleus from the patient. So now we fertilize it in vitro using the sperm, and after in vitro fertilization, we can put it back into a woman and make her carry the pregnancy. This is maternal spindle transfer. The next method is called pronuclear transfer. Here we fertilize the egg before the transfer happens. That is why it's called the pronuclear transfer. Pronucleus means it just immediately after the fertilization happens between the DNA from the father and the DNA from the mother, that nucleus is called pronucleus. So once that happens, I take it out and put it in the healthy cell from the donor. Now, notice that I also fertilize the donor cell. Why do I do this? Because this nucleus is fertilized. I can't put a fertilized nucleus in an unfertilized egg. So I have to fertilize the egg also. So once I fertilize both the eggs, I take the nucleus, I transfer it to the donor cell. So now I have healthy mitochondria and a healthy fertilized nucleus. Then I transfer it to the mother and let her carry the pregnancy. This is how mitochondrial donor therapy is done. So you can compare these two technologies, these two methods of transfer side by side. On the left is the maternal spindle transfer, on the right is the pronuclear transfer. So as you can imagine, this technology has a lot of issues, and you can imagine the number of uh, legal issues that might arise for having three parents. The word parent is in quotes because they're not exactly true parents. We are only sharing mitochondrial DNA from the donor woman, but the rest of the DNA is from the mother and the original father. So the legal issues could be, like parentage or ownership of a child, or all the other issues that are pretty prevalent right now in surrogacy and assistive reproductive technology. Those sort of issues will arise even with this technology. Another issue is using this technology even without having a mitochondrial defect. So let us assume a woman who does not want babies that look like her. Maybe she wants a baby that that is from another culture. Maybe she wants a baby that has blue eyes. Maybe she wants babies that has a lighter skin tone. What she can do is get DNA from another person, right? And she can remove the DNA from her own egg and put it in her egg and fertilize it with her husband's DNA, and can carry to pregnancy. Let me go to this scenario over here. So let us say this couple is in India. They don't like black hair. They want blonde hair. So what they do, they recruit some woman with blonde hair, right? And they want her DNA. So they would just, instead of throwing away the DNA, right? They would just take her DNA and transfer her DNA inside this woman's egg, and you know fertilize it with the sperm and carry to pregnancy. You will have a baby that does not even look like you. It will look like some other uh, a race or some other culture, so on. uh, so those are the issues with having this technology and using it for reasons other than a defect. And you have the same issues with designer babies, right? We have already seen what CRISPR-Cas9 technology could do. Uh, Basically, for those of you who don't know what CRISPR-Cas9 technology, it's similar to your copy paste or cut paste command in your computer. You're basically going to the DNA, cutting out the things that you don't want, and pasting the DNA sequence that you actually want. So you can change the color of your eyes, color of your skin, color of everything, depending on the complexity of the character that you are aiming for. But CRISPR-Cas9 technology is a very highly potent technology to to uh, correct the mistakes of mutations and improve the conditions of patients. This can also be used to do a lot of other changes in embryos. So here is where the concept of having a technology without any regulatory issue uh, might throw up a lot of ethical issues. And again, in India, we just have introduced the Assistive Reproductive Technology Bill 2020. Uh, it might not it might not have been passed yet. It has not been passed yet as of December 2020. But even in Assistive Reproductive Technology Bill, there is no mention of no uh, who is who has to approve of such a uh, procedure even without having a defect. So if the mother and father are perfectly healthy but they still want to have a baby with other characteristics, can they do it with assistive reproductive technology? So those sort of issues have not been clear. So there are a couple of other issues that might arise as the technology is being uh, used on a wider scale. So that is about mitochondrial DNA technology and its issues. I hope you all learned a few things about mitochondrial DNA, and I hope you can leave a comment about any doubts if you have. Thank you.